First of all, thank you everyone for coming to my dissertation defense today, entitled Characterizing Lignin Phenotypes of Potential Bioenergy Crops Towards Elucidation of Novel Candidate Genes through Genome-Wide Association Study. And I'm just stop my video just to help my precious computer. So yeah, we'll jump right into it. As a brief outline of what I'll be talking about today, be covering some introduction, background, and methodology just to calibrate everyone, make sure everyone understands is on the same page. Some of the concepts I'll be talking about in my work associated with lignin characterization towards genome-wide association study in both switchgrass and poplar. Talk a little bit about some additional characterization work that came out of these two studies. I have a handful of other works that I'll briefly summarize towards the end, and then conclude with just some overall high-level conclusions, recommendations for future work, and then open it up to questions and answers. So we'll jump right into introduction, the need for biofuels. So as you probably know, fossil fuels are a major contributor to the release of greenhouse gas emissions. And today, fossil fuels account for about 80% of total energy consumed worldwide. Now, a major consumer of fossil fuels is the transportation sector, which accounts for about 58% of this consumption and is responsible for about 25% of CO2 emissions. And on top of that, the greenhouse gas emissions from the transportation sector has been increasing at a faster rate than any other sector. And as you can see from a press release just over the summer, the EPA has actually increased the quota for the blending of renewable fuels. So we have a motivation and a need to develop and implement biofuels. Now, over the past few years, the focus of biofuels has been shifting to applications like biodiesel, maritime, and aviation fuel. And so there have been some, developments recently in these areas. For instance, ExxonMobil just recently completed a sea trial using marine biofuel oil, which reduced CO2 emissions by up to 40%. And perhaps more importantly, was almost a drop-in replacement, which is going to really allow them to make substantial progress towards their carbon reduction goals. Another area that biofuels is poised to address is the aviation industry, which generates about 2.5% of all current CO2 emissions. And the aviation fuel demand is expected to double by 2050. And this is an area where other power sources like batteries is just doesn't possess the necessary energy density. So aviation fuel is going to continue to be liquid fuel for the foreseeable future. Now, biofuels obviously are going to come from biomass. Biomass is a very broad term. So for this presentation, when I say biomass, I am referring to lignocellulosic biomass, which is called as such because it contains lignin, cellulose, and hemicellulose. And we'll be focusing primarily on the lignin fraction here. Lignin is very important, accounts for 25% of the cell wall, plus or minus, depending on species. And it plays an important plays a role in many important biological functions, such as water transport. It provides strength and rigidity to the cell wall and into plants. And it's often described as the glue that holds the cell wall together. Now, lignin is usually described as an irregular or complex biopolymer, but does have kind of three basic building blocks, three basic monomers or monolignals as they're referred to: syringyl or S unit, guaiacyl or G unit, and then the p-hydroxyphenyl or this H unit here, which differ only in the degree of methoxylation on the ring, as you see. And from this table here, you'll notice that different species will contain different ratios of these S, G, and H monolignals. And then you'll also might notice that this H unit is typically less abundant than the S and the G units. So a lot of times lignin is just classified by the ratio of this S monomer to this G monomer, which we call S to G ratio. And you'll see from this diagram over here, a hardwood like a poplar will contain about a 2-to-1 S to G units, 2-to-1 S to G ratio. Monocots grasses like an alfalfa will typically contain 2-to-1 G units, lower amount of H units, and each can be decorated with a number of non-canonical monolignals. For instance, poplar might be decorated with a p-hydroxybenzoic acid. Switchgrass monocots will be decorated with a p-coumaric acid or a ferulic acid, which participates in cross-linking between the lignin and the hemicellulose fractions. And the other thing that's really neat about lignin is it'll incorporate lots of non-canonical monolignals. For instance, switchgrass, other herbaceous species contain this tricin species, which is actually not part of the lignin biosynthesis pathway at all. It's actually a flavonoid, comes to us through a different pathway. And so main takeaway here is lignin exhibits a high degree of variability and plasticity. So you might be asking at this point, well, why do we care so much about lignin? Well, in this concept of the biorefinery, right, which parallels, petroleum refinery, is we want to take in a biomass feedstock and we want to separate the polysaccharide, cellulose and hemicellulose polysaccharide fraction from the lignin fraction, so we can use each to its optimal end use there. But lignin makes this separation process difficult, and so there are a few different strategies to deal with lignin in the separation process. The first is what you might call a traditional biorefining strategy, which you'll probably be familiar with from like pulp and paper mills, where they use a pre-treatment, usually a chemical thermal pre-treatment, to disrupt the cell wall to gain access to the polysaccharides. Now, like I said a minute ago, lignin is kind of the glue that holds the cell wall together, so it makes this pre-treatment usually ineffective. But there are certain aspects of lignin that will improve the efficiency of the pre-treatment or reduce the efficiency of the pre-treatment. So an active area of research is, can we tailor or modify the lignin in the plant to make this deconstruction process more efficient, so you can use lower chemical and thermal inputs to more efficiently deconstruct the cell wall and gain access to those polysaccharides. Usually during this pre-treatment process, the lignin that comes out is highly condensed, meaning lots of carbon-carbon linkages, and is therefore usually pretty hard to utilize after the fact. What's become more of a focus over the past few years is this lignin first strategy. And really all this is you're in the separation process, you're paying very close attention to and designing your process in a way that the lignin that you get out has a very specific structure that can be utilized in one of these many value-added applications, such as 3D printing. And then kind of the poster child for this right now, very trendy, is to look at upgrading lignin into jet fuel or sustainable aviation fuel for the application that I mentioned earlier. Okay. So now that you know that lignin structure is both highly variable and very important, you might be asking, well, how do we know what the lignin structure looks like? How do we characterize it? We do this today. I've highlighted a couple of common methods that are widely used today. PYMBMS, thioacidolysis, and HSQC NMR are three common methods you'll see in the literature. I'll be talking mainly about HSQC NMR for lignin characterization, and most of you will probably know NMR, nuclear magnetic resonance, and then HSQC, heteronuclear single quantum coherence, which is the specific experiment that we run to analyze lignin. Now, this does come with a few drawbacks. Number one is that it's just a very time and labor-intensive process. It usually takes us about 2 weeks to get an HSQC NMR spectrum on biomass, and that's if we receive it as a knife milled, harvested biomass up front. But the advantage is it gives us a very well-resolved spectra in 2 dimensions, which allows us to really quantify or semi-quantitatively quantify many aspects of lignin structure all in the same spectra. So it just gives us a ton of information that these other techniques usually don't give you. And at the end of the day, what we're wanting to do, the end goal here, is to incorporate this lignin structural data in a genome-wide association study. So a genome-wide association study, the simplest way to describe it is a genotype to phenotype analysis. And to give you just an idea of the workflow, we start with a diverse collection of a feedstock. We do the phenotyping, which is the HSQC NMR. We do the genotyping. And so this is basically DNA sequencing, which is done by the Joint Genome Institute, which is a Department of Energy Bioenergy Science Center. And then the GWAS is basically just a fancy statistical association, fancy statistical model, which will make associations between the genome and the lignin structure. And this is done in a number of different ways, or can be done in a number of different ways. But what it does is it basically identifies genes that may be influencing lignin structure, which will give us some new insight on how lignification occurs. And so as I come to the end of this introduction section, I just want to state a couple of research objectives. Number one is to examine the variability of lignin structure in poplar and switchgrass. Number two is deep phenotype the lignin structure of the poplar and switchgrass to provide the most extensive library of lignin structural properties to date. Number three is to utilize genotype to phenotype or GWAS analyses to identify genes likely to influence lignin and or cell wall traits. And then finally, we want to utilize phenotyping results to develop new insights of cell wall architecture. So like I said, I did work on both switchgrass and poplar. I'm going to start with switchgrass. So to briefly introduce switchgrass, it's a diverse C4 grass, broadly adapted to many different regions of North America, often classified into upland, lowland, or coastal ecotypes, depending on where it's adapted. It has pretty low agricultural requirements while also having fairly high yields. The switchgrass population that we worked with in this study was propagated and grown at Iron Horse Farm in Watkinsville, Georgia, by our colleagues at the University of Georgia. And then this population was harvested in December 2019. We were able to harvest 308 unique genotypes from this field site. So at some point, I usually get the question, what about your NMR measurements? How do you know they're reliable, repeatable, verifiable? So I've inserted this slide here to go ahead and answer some of those questions. So number one is we verified HSQC NMR measurement in a couple of ways. Number one is we have a standard reference material, which in this case happens to be poplar, that we run in triplicate at least each year to ensure that we're getting consistent, repeatable results. And as you can see, just to highlight the S/G ratio from the past 2 years, we're seeing an average, again, of 3 replicates of 1.40 and 1.45 with a standard deviation of 0.05 and 0.03 year over year. The other thing that we did, getting back to the switchgrass population, is we actually ran a couple of technical replicates of several genotypes. And I'm highlighting this AP13 genotype since this is kind of what they consider the standard genotype for the switchgrass population. And as you can see, we ran independent technical replicates in June 2020 and March 2021. And then as you can see, just the variation from these two measurements, independent measurements, 9 months apart, matches very closely our year over year repeatability. And then NMR is a little special in the fact that the lignin structure itself can potentially lead to some bias if you're not careful. But to ensure that we're not seeing that the structure of lignin is not drastically impacting the NMR measurement, we took a sample with a high S/G ratio, kind of a mid-range S/G ratio, and then a low S/G ratio. And we typically only run one replicate per sample mainly for time purposes. But for these samples, we went back and we basically reran these in triplicate. And as you can see, for both the high, mid, and low S/G ratio, all three of these original measurements were within plus or minus 1 standard deviation of the replicate measurements, which gives us confidence that our lignin structure is not biasing the NMR measurements. And then finally, if you present HSQC NMR measurements to people who characterize lignin, one of the things that they like to say is, well, HSQC NMR is not fully quantitative. So have you verified your NMR measurements through quantitative means. And so for this, we selected a subset of about 50 samples, tried to span the whole range of the S to G ratio across the population. And we sent these samples to our colleagues at the University of Missouri, who analyzed the S to G ratio of these samples by thioacidolysis, which is a GCMS-based technique where they use an internal standard, very quantitative. And as you can see, our NMR S to G ratio and their thioacidolysis S to G ratio very well correlated, and with a correlation coefficient of 0.76, which is on par with what's reported in the literature. So all of this just to say we have a lot of confidence that our S to G and really our HSQC measurements are verifiable and reproducible. And so to take a closer look at the switchgrass spectra, again, HSQC NMR is a two-dimensional NMR technique. So along the bottom here, we have our proton shifts. On our y-axis here, we have our carbon shifts, and we get cross peaks at locations where a carbon and proton are J-coupled there. And so, for instance, this cross peak will correspond to the syringeal unit here. This cross peak will correspond to this G unit here, H unit. Again, in switchgrass, it's decorated with coumarates, p-coumaric acid, ferrolates here, and then that tricin unit that we talked about earlier. And over here, this is called the aliphatic region, because the aliphatic chains show up in this region. And so we can get a picture of what kind of linkages bind these monomers, these monolignals together. So for instance, this peak right here, it corresponds to this beta O4 linkage, right? Alpha beta O4. And then this cross peak corresponds to the beta 5, this small one here to the beta beta, and this one here to the dibenzodioxysin linkage. And as a side note, even though we do isolate the lignin through an enzymatic hydrolysis with some enzymes, you can see we still have some residual xylan here in the spectra. And so we did HSQC NMR on 308 unique switchgrass genotypes, and then integrated all the signals I showed you on the previous slide. I think probably a few more, because I integrated 22, volume integrated 22 signals from each NMR spectra. And as you can see, we're primarily G lignin, G units, with about a 2-to-1 ratio to S units. We have a fair amount of p-coumaric acid in the lignin, and then the beta O4 interunit linkage is our dominant interunit linkage. And then I've highlighted here just some overall population statistics, just to look at different, aspects, correlations, to make sure that we're seeing kind of what we expect. For instance, one of the correlations is pretty well established, is between the S to G ratio in this beta O4 linkage. And as you can see, we're getting a correlation coefficient of 0.41, 3 stars, meaning they're highly correlated. So those are the things that we like to see that give us confidence that we're reporting things that are consistent with the literature. So again, what we want to do is we want to take this NMR data and we want to utilize it in a genome-wide association study. And remember, a genome-wide association study is a big, fancy statistical model. And so for switchgrass, we used 4 GWAS methods, 1 single locus method and 3 multi-locus methods. Each one of these methods considered 4.5 million single nucleotide polymorphisms, or SNPs, and those are literally single nucleotides on the genome. And between these 4 GWAS methods, they identified 297 gene associations across 12 lignin phenotypes. And just to give you an idea of what you're looking at here, this is called a Manhattan plot. We have our genome chromosome position here on the x-axis. We've got a significance level here on the y-axis. And each one of these are all individual little dots, which kind of gets lost in the fact that all of these look like solid blocks, but they look like solid blocks because we've got 4.5 million of these little dots represented in here. And as you can see, we've got our significance levels drawn here in these green lines, and we're really looking for these SNPs that are above these significance levels. Those are the genes that we're really interested in. And so when we look at those genes that are identified, we noticed a couple things right off the bat. And again, this is pulling from a list of 297 genes identified by the GWAS, but we notice a couple things right off the bat. The first is that we're seeing a couple of genes that we know for a fact influence lignin and cell wall biosynthesis. Know for a fact is in there is strong literature evidence, it's discussed very well accepted in the literature, that these genes are proven to influence lignin and cell wall biosynthesis. For instance, this MIB domain protein 4, this Werke transcription factor, this NAC transcription factor 35, all very well documented to control lignin and cell wall biosynthesis. And that's really one of the keys or one of the challenges with GWAS is answering the question, how do you know you're getting good authentic hits? This is one way is we're identifying putative cell wall regulators already. And so there are additional ways to identify new genes from GWAS that might be influencing lignin or cell wall biosynthesis. What we did here is a functional partitioning of gene clusters, where genes with related functions are classified or clustered here, represented by the same color clusters. So for instance, if you look at this this pink cluster right here, you'll get a list of about 15 genes, all with similar functions. And if you'll notice, this pink cluster right here contains this MIB domain 4, this NAC 35, and this Werke transcription factor that we talked about on the last slide, that are putative cell wall biosynthesis regulators. And this is represented in this graphic here from a review from a postdoc that I work closely with. And you can see Werke 12, that's this transcription factor here, very high level cell wall regulator. This MIB4, very high level cell wall regulator, and I couldn't find a graphic that also included this NAC 35. But again, we know that these three are high level cell wall regulators. So now all of a sudden, this group associated 12 genes that are predicted, that have the same function or predicted to have the same function, now become very interesting, very high priority candidate genes to look at. So conclusions for the switchgrass work, again, we did HSQC NMR, 22 lignin signals from each spectrum from 308 switchgrass genotypes. We identified 297 genes across 12 lignin phenotypes. Functional partitioning helped prioritize genes identified by GWAS. And then ultimately, the next step of what we want to do is do some functional validation, where we take some of these prioritized genes, transform them into mutant lines, and do some characterization to prove that, or at least evaluate that these genes are doing what we are predicting that they do. And at this point, I usually get a question of, what are you shooting for? What's your target? What are you trying to achieve lignin structure wise? And to answer that, I say, well, this is really still an unsupervised method where we're not going after anything specifically, but this is what we have right now as our ability to control lignin and cell wall structure. And this is what we're trying to obtain. This is again, unsupervised. We're trying to find new knobs to turn to control lignin and cell wall structure for either improved performance during pre-treatment or lignin first bioprocessing and lignin valorization. So that was switchgrass. I'll get into poplar now, and poplar is going to be structured very similar. So poplar, benefits of poplar as a bioenergy feedstock. Number one, it's very fast growing, produces very high yield, it's stress tolerant, has a high cellulose, low lignin content, and grows on marginal land, which can help negate the food versus fuel argument of utilizing prime farmland for non-food sources. So for this work, these poplar trees, Populus trichocarpa, they were all grown in Corvallis, Oregon at a common garden field site. These are all natural variants. And to take a look at the poplar HSQC NMR spectra, you'll notice a lot of similarities. We have our S lignin here, our G units here, H unit is fairly small. In switchgrass, we had the p-coumaric acid peak in this area, whereas poplar, we have the PB, the p-hydroxybenzoic acid peak here. And then lignin, poplar lignin has a lot of the same interunit linkages, the beta O4, beta beta, beta 5, but it has this beta 1 alpha O alpha type linkage instead of the dibenzodioxazine that the switchgrass had. So some similarities, some differences. So we, again, looked at the lignin structure from 409 unique genotypes and volume integrated 12 peaks, 12 cross peaks from each of the spectra. And we had an S to G ratio of about 2.7, right? So about twice as many S units as G units. And then beta O4 was our predominant inter-unit linkage again. And then also with poplar, plotting some population statistics to see if anything stood out to us. Well, in this case, there was something that stood out to us, and that was that the population distribution of PB, p-hydroxybenzoate, was not normally distributed. Now, everything, all the phenotypes from the switchgrass GWAS, they were all normally distributed, and everything from the poplar except this PB unit here was normally distributed. But PB follows more of a log normal distribution, which was really kind of perplexing as to why that would be the one phenotype that wasn't normally distributed. And then when we plotted that against the S to G ratio, right, we noticed a pretty strong negative correlation, especially in this kind of lower half of the S to G ratio, which is also peculiar for a number of reasons. Number one is that PB is almost exclusively attached to S units. So you would expect as your proportion of S units goes up, your abundance of PB would also go up. But we're actually seeing the exact opposite trend here. And we started questioning, well, why does that occur? And the reason is basically we don't know. There's really not a lot that's known about p-hydroxybenzoate. We know that it's linked to S units, but the biological implications unknown. We don't know what function it plays, what importance it plays, and then it may impact recalcitrance. It may not. I think there are publications with evidence on both sides of that fence. But just to demonstrate how much we don't know about this unit, we don't even know the full biosynthesis pathway yet. As you can see here, there have been new genes discovered just in the past year or two that control p-hydroxybenzoate levels in poplar lignin. So we are still discovering new things about the p-hydroxybenzoate unit currently. And these are the kinds of things that we're hoping that GWAS will give us some insight towards. So poplar GWAS results, these are our Manhattan plots for the poplar GWAS. For this, we use the GEMMA method, which is a multilinear mixed model. So for each phenotype, this model considered just over 8.3 million SNPs, which is almost twice as many SNPs as the switchgrass. And we identified 756 genes, including several putative lignin biosynthesis genes. And these are actually in the lignin biosynthesis pathway at all. So we know we're getting associations with known lignin biosynthesis genes. The other thing that we did is we looked at some gene ontology, and basically what gene ontology is each gene is assigned kind of a categorical function, high level. And then if you look at the gene ontology report, as we get more discriminatory with our higher significance levels, we're getting a higher enrichment or higher proportion of cell wall related genes, which again is just giving us some confidence that our GWAS results are returning good associations, promising associations. And so for the poplar GWAS, we actually, instead of functional partitioning like we did for the switchgrass GWAS, we did more like a ranking system where we ranked our genes based on a number of things, number of factors such as strength of association or p-value, connectivity with SNPs, co-expression with phenylpropanoid genes, and basically combined all these into a ranking system to rank and prioritize genes. And as you can see, just kind of a sample here, but we also with switchgrass, we identified some MIB genes, or at least as putative biosynthesis, cell wall biosynthesis regulators. Here in poplar, you see we also identified some MIB domain proteins, MIB 79, 105, 50, and that's just because MIB is a huge class of transcription factors involved in many biological processes. And so while these have not currently been implemented in lignin or cell wall biosynthesis, it's not outside the realm of possibility that additional MIB domain proteins could be influencing lignin biosynthesis. And so, but yeah, so again, this is just a small sampling. I think we put a list together of like 50 high priority genes in our publication that we think are worth pursuing. But yeah, so conclusions for the poplar: HSQC NMR for 12 lignin traits, 409 unique poplar genotypes. Remember that the PB unit was negatively correlated with the S/G ratio and was log normally distributed across the population. And the GWAS identified about 750 new genetic associations with lignin biosynthesis. And again, ultimately, what we would like to do here is do some functional characterization where we transform these high priority candidate genes into mutant lines and do some characterization of the cell wall. And so now that we have 300 switchgrass and 400 poplar samples, genotypes characterized, we wanted to look at a few things that kind of stem from these results. And so I've divided this section into two parts. And the first part is we wanted to look at samples with differential molybdenum content. And I haven't mentioned molybdenum up till now, so let me give you a little background on why we're interested in molybdenum specifically. So again, poplar GWAS provided some really unprecedented insight into lignin structure. One of the genetic associations that the poplar GWAS identified was between this beta-beta linkage here and a molybdate transporter MOT1. And MOT1 is very closely associated with molybdenum content. Now that we actually have the genotypes that were grown in Corvallis that I analyzed for the GWAS study, these are actually clonally replicated in another field site in Clascony. And these have previously been sampled for molybdenum content. And so what we wanted to do is basically sample genotypes, clones from the Clascony site with known molybdenum content and see if they show us differences in the cell wall structure as predicted by this GWAS association. So molybdenum levels in natural variant poplar. So if you look at the molybdenum levels in the Clascony population, we identified MOT1. So this is the Arabidopsis, MOT1 and Arabidopsis. And there are actually two populus genes that have a similar level of homology, both highly expressed in the roots, which is to be expected. And the other thing, when we looked at molybdenum content in across the Clascony population, we noticed that the molybdenum content actually was not normally distributed. It followed more of a log normal distribution, which, we've seen something like that before, right? And so as a kind of a, headline spoiler, but we sampled 3 high molybdenum content trees and 3 low molybdenum content trees to look at for analysis to see if we could find differences in the cell wall. And from the HSQC NMR spectra, we actually did see some differences right away. This high molybdenum genotype actually had very low PB content, about right at just above background levels. And then this low molybdenum content genotype here actually had very high PB content, and you can see a difference in the S/G ratios too, right? So had a 2.47 versus a 1.38. And then if you remember for one of my previous slides, we have an HSQC NMR error measurement of around 0.03 to 0.05. So this is actually very, very stark difference, very measurable. And as I showed in the previous slide, we did have that inverse correlation between PB and S/G ratio that we've seen before. And in addition to just doing the HSQC and MR, we also did a compositional analysis where we looked at lignin content, glucose content, and xylose content by the traditional two-step acid hydrolysis method. And then we did notice, some trends here. For instance, in lignin, you had one of your low molybdenum samples had, very high lignin content, and then another one had at least higher lignin content than two of your high molybdenum samples. Notice some difference down here in the xylose content between low and high samples. And as you can see here, there's may not be kind of a smoking gun as far as these trends are concerned. But remember, again, these are natural variants, and so as a kind of proxy of using molybdenum content as a proxy for MOT1 and just natural variants, these that we're seeing some trends here is actually giving us some confidence that molybdenum and MOT1 may be playing a role in lignification. And so the other thing that we wanted to do was dig a little deeper into this PB content and its effect on the cell wall. So remember, PB has been hypothesized to impact cell wall structure and sugar release, and PB itself actually has a value as a platform chemical. The example that I usually use for this is there's a patent to manufacture acetaminophen with PB as a starting material. So other lignin analytical techniques usually fail to quantify PB. And then the other thing is, as we saw, PB is correlated with S-to-G ratio. So it's difficult to decouple from other cell wall properties, right? So if you have two samples with different PB contents, they're probably going to have different S-to-G ratios. And so you start to question, hey, does differences that I'm seeing come from differences in S-to-G ratio or difference in the PB content? So what I wanted to do here is since we have a library of 400 plus genotypes with lignin fully characterized by HSQC and MR, what I wanted to do is strategically select some samples that had different PB content but similar everything else. And so I dug through my library of 400 samples, and I was able to find a couple of samples which met this criteria, at least as close as you could reasonably expect, given all the natural variation in lignin. But for instance, I have this set of paired samples here, difference of PB just over 10% with an S-to-G ratio around 2, and then these two samples here, S-to-G ratio around 2.8, difference in PB content also just over 10%. And now I mentioned this before, but HSQC NMR is considered semi-quantitative, and for traits like the S-to-G ratio, that's it's really not a huge concern. But for p-hydroxybenzoates, which is present as an ester-linked pendant unit, it has a longer relaxation time since it's not part of the lignin backbone. And what that means, it can be easily overestimated by NMR. So the first thing that I wanted to do was to quantitatively evaluate the PB levels in all of these samples, and do that by a mild alkaline hydrolysis followed by HPLC. And as you can see from this set of paired samples here, we did have differential PB content between the two samples, though maybe not quite as drastic as NMR would lead you to believe. And this is not totally unexpected. We had a publication earlier this year where we did HPLC PB determination along with the NMR PB determination, and got a very good correlation between the two measurements. Although if you'll notice from the scale, NMR returns a much higher measurement than the HPLC. HPLC usually puts maximum PB content around like 2.5% is usually what you'll see there. In addition to the HSQC and the HPLC, we also did a compositional analysis where we looked at glucose, lignin, and xylose levels in the cell wall of each genotype. And you'll notice for this low SG paired samples, we did see some differences in the glucose, lignin, and xylose levels. And then in this pair, we did see some differences in the glucose levels, which we need to account for when we do our sugar release test. And then the other thing that we know can impact sugar release cell wall structure is crystallinity. So we did solid state NMR on each of these samples. These two peaks right here are the crystalline and amorphous peak of the C4 carbon of cellulose. And as you can see, paired samples here, the crystallinity is very similar between each set of samples. So we took these samples and subjected them to a sugar release test, added enzymes based on the glucose levels that I presented on the previous slide. And when we did our sugar release test, we noticed a couple things. Number one is this orange set of samples here is the high S to G ratio, which gives you better sugar release than the low S to G samples, which is expected. But you'll notice in this pair of samples, the low PB content gives you a higher sugar release, whereas in this green pair of samples, your low PB content gives you a slightly lower sugar release. And that trend is consistent between glucose and xylose. So just from these two pairs of samples, just from a standard sugar release test, it looks like PB levels are not going to be especially correlated or influence the sugar release of biomass. And so conclusions, again, this was kind of a two-part section, but number one is we wanted to look at the MOT1, potential impact of MOT1 candidate gene by using molybdenum content as a proxy, found differential PB, S to G, some trends in lignin and xylose levels that give us some evidence that there may be some molybdenum involvement in lignin biosynthesis and populace. And then PB characterization, again, does not appear to be influencing cell wall architecture, does not appear to be influencing sugar release. And if you wanted a positive takeaway from this, again, PB is a potentially valuable as a platform chemical. PB could, if you found a way to increase PB and poplar, you could potentially, as far as we can tell, increase PB levels without a significant penalty. So I wanted to give a quick summary of my some of the additional works and publications during my time here. So first of all, we looked at some lignin from leaves of populace from a field site that half the field site was irrigated, the other half experienced drought, and we were hoping to find some differences in lignin structure between these two treatments. And we didn't find any consistent trends, but what we did find was actually really interesting as far as some unexpected lignin structures. For instance, we found just a ton of condensed S lignin, and some of the samples in the beta-O4 linkage gave us a cross peak in two different locations in the HSQC spectrum, which corresponds actually to two different conformations of the beta-O4 linkage, which was unusual. You don't see both of these. It's usually just the erythro, I believe, in the poplar stem lignin. Another thing that we looked at is we had a field site where poplar genotypes, our population there was infected with a septoria fungus. And so we looked at the healthy and infected tissue from the septoria infected genotypes, noted some difference by HSQC NMR and the SG ratios and PB content. Looked at these by FTIR, were able to tell some differences, distinguish between healthy and infected there, and then also did some compositional analysis where we noted increased lignin content in the infected tissue. And then finally, we looked at some potential contaminants in the HSQC NMR spectra. Probably the one that's most relevant here is I used a cellulase to digest the polysaccharides for the lignin samples that I ran on HSQC. So I ran this cellulase both by itself and with biomass to determine where cross peaks from the cellulase may overlap. And there are a couple instances where that's the case. And so that's for future reference as you're going through to recognize that if your sample is potentially contaminated, you could potentially have these cross peaks in there, which could bias your interpretation of the NMR spectra. And so just finish up here with conclusions and future work. So number one, HSQC NMR measurement of lignin is reliable, repeatable, and capable of elucidating structural variability associated with lignin. HSQC NMR phenotyping provided us new insights towards cell wall architecture and enabled the GWAS to detect associations with those potential causal alleles. And one of the major takeaways from this work is we're coming away with a plus or minus, 1,000 evidence for about 1,000 new genes for cell wall formation. And then ultimately, like I said, the goal, what we want to do to really push this over the edge is to functionally characterize these genes, these high priority candidate genes, which could reveal novel genetic mechanisms underlying cell wall biosynthesis. So recommendations for future work, again, functional characterization, both for switchgrass and for poplar. Number two is, again, we've got a library now of 300 switchgrass genotypes and 400 plus poplar phenotypes for the HSQC NMR. And so we can really actually start expanding on this library, putting together, adding to it compositional lignin cellulose, hemicellulose content, start looking at crystallinity, molecular weight, hydroxyl content, whatever the case may be, but really start selecting samples, adding to the library, and really start looking for some new or some additional relationships that we didn't know or that we don't know are there. And then finally, as I kind of mentioned in the introduction, one of the I say new, it's not really new, but areas of research is how do you take this lignin and utilize it for a value added application. And so one potential application is in sunscreen, right? So lignin is a good UV absorber and could be a good non-toxic and environmentally friendly UV absorber for sunscreen. And again, we've got a library of 700 lignin samples, which could all be evaluated to see what effects the structure has on UV absorption in applications such as sunscreen. And then finally, want to just leave quickly, post the list of publications that I've had during my time here. And then finally, I'd like to acknowledge all my group members, colleagues, collaborators who made all of this work possible. And then would like to acknowledge my committee members for being here today and reading, perhaps almost more impressive is reading a 200-page dissertation. And so yes, I'd like to thank everyone there. And with that, I'll say thank you and then open it up for Q&A.